studies. I'll start with an example of this. Cognitive neuroscience has had a particularly useful role to play in understanding the mechanisms of existing mental health treatments. So thanks a large part to study in animal models, we have an excellent understanding of the biological mechanisms of psychiatric medication. But this exemplifies a typical silo in psychiatry. Medication represents only half of our treatment arsenal, and you can't do CBT or other psychological therapies in animals. So until recently, we had very little idea of those biological mechanisms and how they might compare to antidepressants, which could help us understand why some patients respond better to one than the other. So here I'm showing some results from a recent large-scale reanalysis of many neuroimaging data sets that I conducted. These were all patients who lay in the scanner and did some kind of emotion processing task before and after either a course of antidepressant drugs, which is in the top row in green, or a course of psychological therapy of various different kinds, which is in the orange on the bottom. And what you can see is that the brain regions altered by these two different treatments are anatomically totally distinct. And I tried this in a number of different ways statistically, but they were really totally separate. This was quite interesting because you might think that that explains a little bit of why some people would respond better to one than the other, because they're anatomically working on slightly different circuits. So you could look at my findings there and you could hypothesize, well, what if we scanned someone's amygdala at baseline and we had an idea of the level of activation there that might correspond with whether or not they'd be someone to respond to antidepressant drugs. So luckily, if you said that, people have tried that, so you're about to have your question answered. Here's a study from Leanne Williams' lab from a few years ago, where she scanned patients before giving them a course of antidepressant drugs. And if you look at the green bars pre-treatment, those patients who responded to antidepressant drugs had profound hypoactivation in the amygdala compared to those who didn't respond. And interestingly, those same patients showed this kind of hyperactivation after treatment, showing that that increase in activation is probably what's driving my result on the left. But if this is true, this could be really useful. You could use amygdala activation as a sort of predictive biomarker. Unfortunately, I think there are a few reasons why you might want to be skeptical about this and other findings, because I think there may have been a sort of step in that translational pathway that I showed you on the last slide that was skipped. For amygdala activation, or any measure, to be used as a predictive biomarker in psychiatry, it has to have particular statistical characteristics. Chief among those is within subject reliability. What I mean by that is, if I scanned everyone on this call today and I identified those of you with the top 10% highest amygdala activation in the group, so according to the trial, the ones who might respond to antidepressants, well, if I scanned you again in an hour or again tomorrow, you had better still be about the top 10% in the group. And this reliability is relative to the rest of the group. The absolute amygdala activation can, of course, change, habituate over time, and so on. But if you want to be identifying people in comparison to others based on that, then relatively that better remain the same over time. You better have a robust and reliable biomarker. So one of the studies we ran a few years ago tested this empirically. We tested both amygdala activation and activation of another putative biomarker region, the subgenual anterior cingulate. And what we found was that they were not reliable at all. This figure is showing on the left a control region, the fusiform face area, which was pretty reliable within the subject, either half an hour later or 2 weeks later. But our three potential predictive biomarkers had really pretty disappointing reliability. None of them met the criteria for excellent reliability, which you would hope any potential biomarker would meet. And this was the case in 3 different emotion processing tasks and different ways of doing the kind of region of interest identification of the amygdala. So nothing seemed to make it better. This and other studies replicating and extending this finding since then have really driven home the message that a potential biomarker must be reliable. And I've taken this concept with me towards my more translational studies. And today I really advocate the inclusion of measures of robustness and reliability in all early stage clinical trials, so you can simultaneously test the existence and the viability of a biomarker. I'll briefly tell you about one of those different neural targets. Across many studies, including my own work, patients with major depression show marked differences in dorsolateral prefrontal cortex activation during working memory. This is an example of the neural activation associated with cold cognitive deficits in depression. That's stuff like impairments in attention, memory, and so on. Unfortunately, these very same aspects of depression might prevent people with depression from engaging in relatively difficult tasks like cognitive behavioral therapy. So a number of years ago, in unmedicated patients with major depression, I worked with the Camden Islington Psychological Therapy Service, part of the NHS, to deliver transcranial direct current stimulation of the prefrontal cortex, preceding each of 8 cognitive behavioral therapy sessions. We hypothesized that this would enhance patients' response to CBT compared to an identically sensation placebo stimulation that we used. The molecular mechanisms of TDCS are now a little clearer, and they're known to be relatively complex. They involve non-neuronal and NMDA-dependent effects. But at the time, our original design was based on this finding in animals that a small amount of electrical stimulation delivered externally, so non-invasively, moves the resting membrane potential of neurons under the electrode towards depolarization, an effect which maintains for at least 90 minutes after stimulation, very useful for a kind of pre-therapeutic intervention like this. Our trial included cognitive and neural assessments and a bunch of stuff I'm not going to tell you about. I just want to tell you about that baseline neural activation that we measured in the DLPFC, like the amygdala was used in previous studies. So the trial took a few years to run, but in the end, as you can tell from the relatively small difference between the orange and green bars representing TDCS and placebo, sham, enhanced CBT respectively, we did not find an augmentative effect of the stimulation on CBT, but it was a really dissatisfying no. It was a P of 0.06. So in fact, a Bayes factor analysis showed that the difference didn't support or refute the null. We now know more about TDCS, and we know that response is extremely variable, but this wasn't known when we designed the study. We, at the time, knew about the variability in response to other types of treatment, antidepressants, psychological therapy, and we thought maybe this type of brain stimulation would be similar. So we used that biomarker approach, extracted activation from the left dorsolateral prefrontal cortex, and we found that it was very strongly related to treatment response. That's the orange line in the figure there. This was specific to the augmented condition, not the placebo or greenish line, although I can see it slopes up a little. That's not significant. So it's a specific predictor of brain stimulation enhanced CBT. Crucially, and unlike amygdala activation, when I tested its within subject reliability, it was just about excellent, an ICC of 0.7, making it a reasonable putative biomarker for people to test in larger trials. After running the trial, I was convinced that cognitive neuroscience could start to address individual differences really head-on in psychiatry. And one of the ways to approach this, and an approach I now take in my body-brain intervention work, is by developing interventions that target transdiagnostic psychiatric dimensions, cognitive metrics that have relevance across disorders. In my first postdoc, I used a well-known sequential decision-making task, which is in the top left corner, and a hybrid reinforcement learning model developed by Nathaniel Daw, which has been reliably used to identify deficits in goal-directed behavior across addictive and compulsive disorders. Our primary outcome was a computational parameter implicated in that deficit. It's called W, but all it does is it shows the degree to which an individual is goal-directed or habitual in their decision-making. Are they just reinforced by a reward that just happened, or do they take into account the broader structure of the task, making a goal-directed decision on every trial? I then developed this dual coil transcranial magnetic stimulation approach to target specific connectivity that we knew was implicated in this task. This was designed with John Rothwell, so we worked to time the TMS pulses precisely to putatively enhance the connectivity from the inferior parietal sulcus to the lateral prefrontal cortex. And we compared this to the exact same dual coil stimulation, but just with a longer timing that seemed physiologically implausible. What we found was that this precisely timed inferior parietal to prefrontal stimulation shifted participants in a goal-directed direction. It made them a little bit less habitual on this task. So this was the beginnings of a potential intervention for a transdiagnostic phenomenon, something you see in lots of different disorders. Since then, my goal has remained the same: identifying well-specified neural and cognitive mechanisms and testing them in experimental medicine studies as putative treatment targets. But for the past 3 years, I have had a privilege of time to spend thinking quite deeply about the processes actually causing mental health disorders. The question I like to think about is, where do symptoms come from? The most important function of the brain is to detect and regulate our body's internal state, keeping us within viable physiological operating ranges, not starving, totally dehydrated, and so on. Studies dating back decades have reported disruptions in how people experiencing mental health disorders process internal bodily signals. So I think disruptions in body-brain interactions might engender poor mental health, even across disorders that look superficially very different, by disrupting different levels of this body-brain processing hierarchy. I think this could explain why many disordered general, environmental, and genetic risk factors for psychopathology exist. It's actually very rare to find a risk factor that's specific to a particular disorder, or even a neural instantiation that's specific to a particular disorder. There are two reasons why I think these interactions are critical in mental health disorders. The first is theoretical motivation. My and many other scientists' work shows that the neural circuits we use to support homeostatic regulation are disrupted across mental health disorders, and theoretical work implicates this disruption in the pathophysiology of essentially all mental health disorders. Formal Bayesian models of homeostatic and interoceptive processes, like the schematic I'm showing here, which comes from class Emma Stefan, provide a testable framework for novel neural or peripheral targets in the domain. But the second reason I think these processes are so central is because of a clinical phenomena, which illustrates the inseparable relationship between our physical bodies and our cognitive processes. About a decade ago, I met a patient with functional paralysis who was being treated at the National Hospital in Queen's Square. She had been in a wheelchair for 2 years. Until I met her, I was unfamiliar with functional disorders. But functional disorders exemplify the ability of the brain to alter bodily experience without the typical pathologies we associate with something like paralysis. Functional symptoms exist across all cultures, and some of their symptoms have been recorded in the earliest medical records. Essentially, I think the ability of the brain to predict and regulate bodily state is where symptoms come from, but it's particularly influential in mental health conditions and conditions that lie on the boundary between neurology and psychiatry. Even in diseases with known pathologies, like multiple sclerosis or arthritis, only about half of a patient's functional impairment can actually be explained by their disease progression. To explain the rest, we need a comprehensive understanding of how our brain predicts, represents, and regulates our body's experience, and also how peripheral physiology influences the brain. This interaction is key to understanding where mental health and mental health disorders come from. I'm going to zoom back in now on some experiments and tell you about those two approaches I'm taking in my current work. The first approach takes this kind of Bayesian model of interoception and asks a really pretty simple question that I don't think had been asked yet. So this model exists to minimize interoceptive prediction errors. That's its hypothesis, that what we do, our goal of interoception is to reduce surprise between what our body what our body's experiencing and what our brain is predicting. And there are basically two ways to reduce this surprise: by changing your prior predictions or by acting on the world or the body until those predictions are fulfilled. What's not clear from these models, and something I am very interested in, is at what level or levels of this hierarchy disruptions in mental health disorders might occur. One possibility is that they occur at a very low level, a disruption in communicating visceral information to the brain. Alternatively, there might be common disruptions in very high-level predictions, those anterior regions like the orbitofrontal cortex, predicting future bodily state. Neuroimaging studies have come up with all sorts. broad regions implicated. But as with many fields, these findings can be a bit unclear, not only because of the diversity of tasks used, but also because many studies only report masked or region of interest-based analyses. So you get this kind of rich club getting richer effect, where if one study reports a particular region, the next study uses an ROI approach of that region, and so on and so forth. Also, as I've already mentioned, I think there is this kind of highly needed shift away from a diagnostic model in the mental health literature and towards more dimensional approaches. So what I decided to do, and this was just over a year ago, was to try to identify a common locus of disruption across interoceptive domains and across various psychiatric disorders. And this might not exist, or it might not have existed, and I wanted to know if it did exist, where it might occur in the interoceptive hierarchy. So to answer this, I ran a neuroimaging meta-analysis in 1,236 people, about 600 patients and controls each. Neuroimaging meta-analysis, for those of you unfamiliar with it, is basically a powerful way to aggregate imaging data across studies to find a common locus of disruption in this case, something that exists across tasks and disorders as a difference between a patient group and a control group. But to run this study, I had to make two decisions that I knew some people in the field would always disagree with me on whatever I chose. So the first decision was what kind of imaging contrast counted as interoceptive. You can see the decision I made here, which is based on Sarah Garfinkel and Hugo Critchley's definition, and it's a pretty general and inclusive definition of interoception. And the second decision I had to make was what counts as a psychiatric group. And there I, in line with my transdiagnostic perspective, didn't only include patients with diagnoses kind of officially diagnosed according to manuals, but also those scoring very high or low on particular mental health symptoms. That could have been another way to identify groups. I pre-registered the study, included a long list of symptom terms. This is all a little bit dull, several imaging terms, and a long list of interoceptive terms. Our inclusion criteria were relatively straightforward, but I want to tell you about the last two because I think what this study can and cannot show relies on these particular inclusion criteria. So to do this type of neuroimaging meta-analysis requires whole brain coordinates, things reported across the whole brain without any kind of special thresholding for certain regions of interest. This is kind of a cool feature of imaging meta-analyses because it means that whatever you find in it, it's a little bit less subject to publication bias than the original papers, because most of the stuff that goes into the meta-analysis is actually in the supplemental material of the original papers. And in many cases in my meta-analysis was actually sent to me, the raw data was sent to me by authors so that I could extract these findings, because usually the headline result is a kind of hypothesis-driven region of interest-based finding in the brain. So in the end, we had this 1,236 participant sample, and our age range ranged from adolescence to adulthood, but I'll show you how that differed in a moment. And in these various studies, there were a few different interoceptive probes. There were 5 that measured neural activation during interoceptive attention, stuff like focus on your stomach, very popular task. 10 measuring neural activation during breathing load or breath hold. 8 measuring neural activation during pain. 5 measuring neural activation during effective or pleasant touch studies. Participants get stroked lightly and pleasantly with a paintbrush. I always think that sounds like the most fun to be a participant in. And 5 that manipulated hunger as an interoceptive probe. This is the analysis procedure that I followed. For every study, I extracted the results, these coordinates, that's our kind of primary result, from a whole brain group contrast, for example, a second level contrast of depressed patients versus controls, from a first level contrast where they were all sitting in the scanner doing an interoception versus exteroception task. Activation likelihood meta-analysis essentially considers these coordinates to be 3D probability distributions for a particular voxel in the brain. Which are then smoothed with a Gaussian kernel inversely proportional to the sample size, reflecting the precision of the estimate. So large sample studies get overweighted compared to very small sample size studies. If you imagine that this happens for every study, here I've just made up. There's say a depression paper and a substance use disorder paper and an anorexia paper. But you can imagine this times there are 306 coordinates in total. Eventually, you get a map reflecting the convergence of all these results, which you then threshold very stringently to find out where in the brain, if anywhere, this convergence is more than you would expect if they were all just activating random bits of the brain, if it more than at chance. If you look at the uncorrected threshold before you look at that, what's activated more than chance, you can see a really diverse set of regions implicated in these interoceptive differences between patients and controls. Some of them you could have guessed, but I think some of them are really a little bit surprising if you look at them all together. But if you use the statistical recommendation from the kind of current papers using this technique, and you threshold it at P of 0.05 family-wise error cluster corrected, you get a very different finding. One region of the brain that survives this very stringent thresholding, the left dorsal mid-insula. This region appears disrupted in almost all the disorders and interoceptive probes I included in the original analysis. Bipolar disorder, anxiety disorders of several sorts, major depression, anorexia, and schizophrenia. And this region is a crucial one in the interoceptive hierarchy. We think the mid-insula result could be a common locus of disruption across psychiatric disorders, disorders with other domain or pathology-specific interoceptive changes elsewhere in the brain, but converging on the mid-insula. But before I tell you about that and kind of speculate a little more, I want to tell you about the robustness of this result. First, I wanted to check if it was driven by the smaller number of adolescent studies we included, because I did mention that our age range went down below 16. Excluding those studies, it made no difference, actually. It strengthened the peak a little bit, but stayed in the exact same location. The second thing I was interested in is whether this disrupted activation was because of hyper or hypo activation, because it actually conflates both. That's what the method does. So to answer this question, I separated each of the 306 coordinates into whether it described hypo or hyper activation in patients compared to controls. In fact, neither group survived stringent cluster correction. But if you look at the unthresholded maps, you can see that the blue results, which is where patients were hypoactive in the mid-insula compared to controls, came out, but not in the hyperactive set of studies. So while they both probably contribute a little bit, I think it's fair to say that the studies reporting hypoactivation in this region were the major contributors to this finding. Next, I was really curious to find out if this mid-insula region I had discovered overlapped with regions that already changed following antidepressant or psychological therapy interventions, if it was like a known mechanism of treatment. So to do this, you can contrast the various coordinates that emerge from two meta-analyses quantitatively. It's a kind of meta-analysis, and you get any conjunction regions or regions significantly different between the two meta-analyses. So first I contrasted this with raw data from the largest meta-analysis of the neural effects of psychological therapy that I could find. The authors kindly shared the data with me. I found no regions of significant overlap, even at an exploratory threshold of P of 0.001. So this mid-insula region, which is here plotted in orange, did not anatomically overlap with changes after psychotherapy once you put any statistical thresholding on there. I wanted to do the exact same approach with antidepressant medication. So I contacted an author, Yina Ma, who'd recently run this great study in molecular psychiatry about the effects of antidepressant drugs, and she sent me her data. But I found the exact same finding. There was no significant overlap at the corrected or even at the exploratory uncorrected threshold between where in the brain changes after antidepressant drugs and where in the brain is different between patients and controls during interoception. And the final thing I was interested in, and this was more from a theoretical than a clinical point of view, was whether this dorsal mid-insula region was already part of the larger known affect circuitry of the brain. The reason this is interesting is because existing interoception theories and emotion theories have suggested that any interoceptive differences in mental health originate from the same neural system responsible for affect and emotion dysregulation in mental health disorders. So I wondered if this mid-insula was actually part of the brain's general affect circuitry, and this was something I could test quantitatively using a similar approach. So I got in touch with Kristen Lindquist and Lisa Feldman-Barrett, who shared with me the largest database of neural activation during affect processing. This is in thousands of people. And I compared it quantitatively with the interoceptive patients versus controls data. Once again, and somewhat surprisingly now, I found no overlap. This suggests that the region of disrupted interoceptive processing in the dorsal mid-insula, in orange here again, does not form part of the brain's affect network, which you can see in green here. It's anatomically separate. In conjunction with the previous comparisons I made with antidepressant drugs and psychological treatments, this suggests that the dorsal mid-insula is a region disrupted in psychiatric disorders not targeted by current treatments, nor does it form part of the network we usually think about when we think about the neural basis of psychopathology. My findings suggest that the dorsal mid-insula might represent a common neural locus of disrupted interoception across psychiatric disorders. What I'm particularly interested in is why this might be the case. And to understand this, I want to briefly mention the neuroanatomy of this region and why this anatomy makes me think it might have a particular role in one process, in computing interoceptive prediction errors. To tell you why I think that, I have to tell you a little bit about the cytoarchitecture of the insula, which is fascinating. It has 3 cytoarchitectonic subregions. This ventral anterior agranular area, which is in green here, a mid-insular dysgranular zone, which is presented in light blue, and a posterior granular area. Human and primate anatomical tracing studies have shown that while the posterior insula projects primarily to posterior superior and middle temporal areas, the anterior insula communicates with inferior frontal regions. And crucially, this dysgranular mid-insula, where my result is, has a special hybrid connectivity pattern to both the anterior and posterior insula, managing to communicate between these frontal and temporal regions connected with those insula subregions. This means that the mid-insula is in this ideal anatomical location to integrate predictions from anterior regions and interoceptive input from posterior regions and calculate prediction errors. I would hypothesize that different disorders show disruptions at various levels of the interoceptive hierarchy, but maybe regardless of where in the interoceptive hierarchy a particular disorder's disruption is found, or is caused by, it has these knock-on effects on interoceptive prediction error signaling, which my study picked up on, coded in this mid-insula region. This idea will be one of the central theories I test over the coming years. The second approach I want to tell you about is totally different. It's a small-scale experimental medicine study looking to discover a new possible target of body-brain processing in psychiatric disorders. And unlike the last one, which looked at brain-to-body connections, this one is really focusing on body-to-brain. The study I'm going to tell you about involves an organ that is occasionally neglected in these body-brain interaction models, the stomach. The pacemaker cells in our stomach spontaneously depolarize and repolarize, causing the smooth muscle to contract rather slowly, about 3 times per minute. But when you're nauseous, this rhythm changes. Even if you're not nauseous, but you just look at some images that are pretty disgusting, they have to be a bit more disgusting than that image of feces, that can evoke gastric dysrhythmias. Incidentally, these very same gastric dysrhythmias during disgust observation also correlate with the amount of activation in the dorsal mid-insula. But what's not known is whether gastric dysrhythmias themselves have an effect on cognition or behavior, which is what we set out to address. One of my key motivations for this study was the relative clinical intractability of pathological disgust. My collaborator and mentor, Tim Dalgleish, describes in his book a police officer who had witnessed a very traumatic homicide. This patient suffered from post-traumatic stress disorder, PTSD, but unlike many patients with PTSD whose disorder originates from a highly fearful situation, hers came from a really horrible and disgusting scene she'd witnessed on the job. Her pathological disgust had profound effects on her life. She lost her job, had to give up her cat. And unlike patients with avoidance behavior that resulted from a fearful situation who can respond quite well to cognitive therapies, she didn't respond to typical psychological interventions like CBT or exposure therapy at all. The idea that pathological disgust is immune to psychological therapy is mirrored in the experimental literature. I'm showing you a cool result from my collaborator, Edwin Dahlmeyer. He found that exposure to even very, very many disgusting images in a row does not cause automatic habituation of avoidance behavior. He was measuring ocular motor avoidance, so unconscious eye movement avoidance of disgusting stimuli. And you can see in the left image, which plots the amount of dwell time that someone's eyes spent on a disgusting image versus a neutral image on the screen, people's eyes looked far preferentially at the neutral images in green than they looked at the disgust images in yellow, and that didn't change over time. There was no habituation. This is quite different than the story in fear, which is on the right, where you can see that almost immediately there's some degree of habituation toward the fearful image, and people start to get curious and look at it, and then by the end of the experiment, people aren't bothering to avoid it at all, which is kind of the basis for exposure therapy. So the combination of this work and the clinical literature made me wonder whether cognitive approaches like exposure were unsuccessful, in part because the visceral changes evoked by disgusting images in the stomach, gastric dysrhythmias, might be provoking or encouraging disgust avoidance. I wanted to test the causal role of gastric state in disgust avoidance, so I used a classic approach from experimental medicine and psychopharmacology to test this idea. You take a drug with a known mechanism and you test whether that particular mechanism affects the aspect of cognition or behavior you're investigating. Here, the drug was the peripheral dopamine agonist called dopamine antagonist, which is a common antiemetic. You can get it over the counter in many countries, although most recently not in the UK. Crucially, from a physiological point of view, what domperidone does is it restores gastric dysrhythmias to normogastria under this particular dose that we used. We recruited young and healthy participants who were required to fast and abstain from any kind of alcohol, caffeine, nicotine, that kind of thing that might interact with the drug. We then conducted a medical screening to make sure they were safe to take this drug. And then before giving them the drug, they did baseline ocular motor avoidance trials. What's being plotted down at the bottom is a histogram of the horizontal eye gaze fixation position. So you can see people's eyes obviously prefer looking at this neutral image of buttons than they prefer looking at this disgusting image of feces. We then did a second medical check to make sure. We gave them the drug. We waited 30 minutes, which is the optimal amount of time for the drug to take effect on an empty stomach. And we did this in a double-blind, randomized within subjects design. So we didn't know who was taking which. And everybody took both, but with an amount of time between the two studies. After the drug, they essentially repeated that baseline task. And they also did something kind of cool, which is like a paid exposure therapy task, where we paid participants, we incentivized exposure to look at the disgusting images over a period of time, and then measured whether this had any effect on their ocular motor avoidance. Now, crucially, everybody responds to this intervention, but under normal circumstances, it has no effect on subsequent ocular motor avoidance. People go right back to avoiding the disgusting image. Briefly, we pre-registered the study. We planned for a largish number, but we actually had to reduce our number because this was supposed to end last summer, so we ended it slightly early in the pandemic. So we ended up not doing any of the individual difference analyses, only our primary analysis, which this was adequately powered to test, given a medium effect size. So we've done this data with two different versions, either with no exclusions or with exclusions, and it doesn't change any of the tests that we conduct. And we've also reported the results in a couple different ways. You can either do it with linear mixed models, which a reviewer wanted, or with the repeated measures ANOVA, but they both confirm these results. So that's just to kind of underline the relative robustness of this finding. We also did both frequentist and Bayesian analyses for all of these. What you can see before we gave participants the drug is essentially a replication of Edwin's results that I showed you earlier. The proportion of dwell time spent looking at the neutral image, which is plotted in green, or the disgusting image in yellow, didn't change after domperidone or obviously after placebo on this kind of basic trial. During the exposure trial, there was also no difference between the conditions. Everybody looked at the disgusting image, earned some money. But as soon as we stopped paying participants, the people who were on placebo went right back to avoiding the disgusting image. That's in the dotted lines here. Whilst the people on domperidone got a little bit closer together. So they stopped avoiding the disgusting image quite as much relative to the neutral image. And this was borne out in our frequentist and our Bayesian analyses and in our various types of models that we conducted. Interestingly, this wasn't borne out in a subjective disgust effect, which was not our primary outcome, but we thought it might change. But this is not that unusual in these kinds of exposure therapy type studies. You can sometimes see effects on fearful behavior, but no effect on someone's subjective perception of fear. That's pretty common. As an aside, I did say that domperidone is a dopamine receptor antagonist. Now, it is essentially and almost exclusively peripheral dopamine receptor antagonist. It doesn't seem to deposit whatsoever in motor, prefrontal, or striatal circuits in the brain. But as a cognitive neuroscientist, particularly one who has worked quite a lot on reward processing, I thought I would be a real idiot to not include a control task and make sure that any effect of this incentivized exposure wasn't due to some effect on reward processing. So that's why we also included a reinforcement learning task, because there have been a bunch of other studies showing that at least brain-based dopamine antagonists affect reinforcement learning. So here was the simple task we used. It's one that's almost identical to the type of task where D2 and D3 receptor antagonists affect behavior on this task, different reinforcement learning parameters that I'll show you in a second. We tested domperidone versus placebo using two different reinforcement learning models. And for the sake of time, I might go a little bit quickly over this. So that the only difference between the two models, for those of you interested in computational modeling, was that one had one learning rate or alpha in a reinforcement learning model, and one had two learning rates, which would be if people were learning better from if people were learning differently from positive and negative outcomes. And in other experiments, sometimes one and sometimes the other wins out. It depends a little bit on the particular paradigm, and also in some of my more recent work on the individual person, but that's definitely an aside. And also we used a softmax to fit the choice temperature parameter, which is very standard. We found no effect of domperidone on reinforcement learning, either in frequentist models testing for the main effect of drug, or more importantly, the interaction between drug and phase, these columns here. And we verified this with Bayesian approaches, which found support for the null model. So this provides pretty reasonable evidence against any central dopaminergic effects on learning and reward that might have driven our main finding. It seems reasonable at this point to infer that the peripheral actions of domperidone, restoration of normogastric activity while viewing disgusting images during exposure, may have reduced subsequent ocular motor avoidance. Sorry, I should have clicked through that. So in summary, we think this is evidence that normalizing gastric state during motivated exposure reduces disgust avoidance. It's interesting that this involves and requires actually engagement with a disgusting stimulus. My future work will build on that result, testing combined psychological exposure therapy and domperidone in future, and trying to replicate our central finding, of course. It's important that our results don't seem to be driven by any typical central dopaminergic effects on learning or reward. So we think this means gastric state plays a causal role in disgust avoidance, which is a new piece of evidence for the effect of the viscera on cognition and behavior. Potentially, domperidone could be tested in future clinical studies to weaken the intractability of pathological disgust. There are a few caveats I wanted to highlight about this study, though of course we can go into more detail in the questions if you want. So first, our study uses only a very specific type of disgust called core disgust, which includes feces, but doesn't include, for example, disgust you might feel about injuries, bodily injuries. That's because there's stronger evidence for the role of gastric dysrhythmias from core disgust, not necessarily in the kind of injury disgust. My current hypothesis is that if it does extend to other types of disgust, it's probably what the literature sometimes calls orogastric disgust, basically like things that make you go, bleh. That category includes some, but not all, types of moral disgust, which is often something people are interested in from this finding. So obviously that might need to be tested. I should also mention, for people interested in the stomach, that there may be gastric state specificity. People under a sated state, because all our participants were basically hungry, might not show this same effect. We had to do that because of the drug, but it could be that in a clinical context, you would basically need people to have an empty stomach for this to work. Finally, I want to be cautious about our null result as well. We found no effect on reinforcement learning for monetary rewards, but who knows, gastric state could affect learning and reward processing for gastric related rewards like food. I think that would not be implausible, so I don't want to rule that out from my findings. Or it could affect other types of behavior, such as vigor, because vigor or effort is known to be affected by some gastric states, such as hunger. So I think there are a lot of directions this work can go in, and I'd be happy to kind of discuss any of them, really. I wanted to end my talk on a little bit of an overview of where my work is going. As a cognitive neuroscientist in the field of mental health, my work falls within loads of different subfields: computational psychiatry, precision medicine, psychopharmacology, and so on. But what all these have in common is the goal of discovering, first of all, new ways of categorizing and understanding disorder, moving beyond diagnostic nosology and towards something that may be a more accurate way of delineating differences between pathologies. But why do this beyond new ways of describing clinical phenomena? Well, in my work, it's to improve our currently limited ability to treat mental health disorders. But if this is the goal, that leaves us with a big translational gap. And to bridge this gap, we must move beyond descriptive models of mental health, scores on a questionnaire or even one single questionnaire measure, to discover neurally grounded models of mental health, linking brain to behavior. As I've said, I think cognitive neuroscience has this unique role to play in this, mapping the relationship between physiology, like inflammation, and mental state, or interventions like drugs or TMS and mental state, or psychological therapy. But this approach requires discovery science and experimental medicine studies, and the hardest studies of all, translational studies, to test theories forwards and backwards in early stage clinical interventions. So that's the end. These are the collaborators I'd like to thank, and all my other colleagues here. And thank you all for your attention. Are there any questions?
Hi, sorry, I was muted. Thank you very much for that talk. That was really good. I'll give people some time to ask questions, but in the meantime, I had my own question. That might be stepping aside. It's more going to an evolutionary maybe reason for this sort of these observations. So say with fear you have habituation that happens and there's none with disgust. Could it be that it's just that disgust is really an indication that there is something very harmful to the body here, and that sort of harm isn't going to go away, so you need to continue avoiding that potential harmful stimulus. And then
oh wait, no, go on. Yeah.
Oh no. And then like if that's the case, then maybe adding the whole effect of domperidone, it only becomes effective once you've actually interacted with a disgusting thing. It's because the association between disgusting and harmful is so strong that you actually need to kind of prove to your brain that it's not actually harmful. There needs to be this sort of interaction there. And that's why it becomes like the domperidone becomes effective. And then if you take it a step further, is that association something that's learned or that you're born with? So at an earlier age, if you test it in children, is it something that's that you find, like, if you find the same effect in children? Like, would you assume that you'd find the same effect in children? Or
Yeah, that last one there's a big debate about. So largely, I mean, I agree with you completely. I think that there is something potentially different about the way that our brain and our body has to deal with disgusting stimuli versus fearful stimuli. And I think some of that relates to the actions that are initiated by those stimuli. So fearful stimuli tend to inspire kind of these fight or flight responses. And I think that those are perhaps more easily overridden via cognitive therapy. So they are important. They're still important for survival. It's just a different process. And interestingly, from a body-brain point of view, they tend to involve cardiovascular effects, unlike disgust, which tends to involve this more gastric system. So I think it's possible that the reason we would have like a gastric basis for disgust is because it gives us backup. If we're not thinking, if we're not cognitively avoiding something, we have this almost kind of like instinctive reaction to things that are disgusting, where we don't have to think, oh, that made me sick last time. There's no kind of cognitive processing needed for some of these really basic responses. I agree that the domperidone result is, it is when we expected it to happen. We expected the exposure therapy to be needed. And the reason for that is because we thought otherwise people won't have engaged with the disgusting stimulus. And in a typical fear exposure context, you actually need, often like in therapy, people hold spiders and things like that. That's what you work up to in order to get over something like spider phobia. So similarly, we thought we had to have people actually engage with the stimulus, not avoid looking at it. Otherwise, it wouldn't take effect. And then to answer your very final thing, in children, all I will say is there is a big debate about it. Some people argue that disgust is essentially innate, that we're born with these kinds of disgust responses, and then sometimes they can extrapolate to other things like moral disgust. But I certainly know researchers who think that even basic forms of disgust are learned.
Yeah. Okay. We have a few questions here. What happens to those people who are kind of, okay, if you transect people who have a transected spinal cord where the periphery is disconnected, like what would you expect to see in those?
In what context? Like clinically, or?
I assume, like, yeah, like how would the results be if there is no communication, I guess, is what they mean.
Okay, I understand that. So certainly in the context of the domperidone study, I guess you would expect it not to work, right? That would be my hypothesis. I think if, for example, if you didn't have a vagus nerve, I would expect it not to work, because ultimately that's how information is being conveyed. I don't imagine some kind of other path that could possibly be. Yeah, so that's what I would expect. I would expect it not to work.
Okay. Great talk. I was wondering whether there are studies linking the metabolic states of patients with psychiatric disorders.
Yes, it depends what you mean by metabolic states. Like there are, so the area where the majority of this research on metabolic states has been done is eating disorders. And there are certainly very interesting studies looking at differences in processing satiety versus hunger in, say, people with anorexia. So they have often, you see this kind of aversive response to satiety, whereas people without anorexia tend to quite like feeling full. And in people with anorexia, there's this maybe learned response that is that makes satiety kind of aversive. Whether that extends to the actual metabolic processes in the stomach is actually something that hasn't been really fully explored to my knowledge.
We have another question. Hello. Amazing talk. In the shown ALE meta-analysis, antidepressant drugs treatment didn't show any overlap with aberrant activation in the mid-insula, but did treatments such as antipsychotics or benzodiazepines show a different result?
Good question. That wasn't in that particular meta-analysis. I personally would expect that they might. you do tend to see results, various kind of peripheral body type results from something like benzodiazepines, more so than with antidepressants. So you would acutely see changes if you gave someone benzodiazepines, and maybe that would affect the minute cell. I don't know, but that's pure speculation. So that particular meta-analysis only included SSRIs and SNRIs, but in future it would be quite exciting to look at it in the context of other treatments as well. I tried really hard to get a brain stimulation meta-analysis, but no one would send me the relevant data.
Okay. I think that's okay. Someone's adding that there is some work on metabolic syndrome and depression. No idea about cause or effect.
Yeah, well, that's actually another interesting point. So if we're talking about metabolic syndrome, that's a separate category. So metabolic syndrome is one of the various physical disorders that is pretty common across various mental health disorders, not even just depression, but those two are particularly comorbid. And there seems to be, my understanding, not specific to metabolic syndrome, but about physical disorders in general and depression, is that there's causality in both directions. People with depression are about 3 times as likely to get diagnosed with some kind of chronic physical disorder. And then it works in the other way around. If you have a physical disorder, you're far more likely to be diagnosed with depression.
Interesting. Well, we don't have any more questions for now. I've attached the link to the Zoom discussion in the chat box. And please join us next time when Manuel Sempere will talk about brain-body interactions in the metabolic nutritional control of puberty. Thank you all very much. Thank you, Dr. Nord, for the talk. And we'll see you soon.
Bye.